Good day, everyone. We will be discuss here today about the fungal diseases in aquaculture. So basically, we will be discuss here about what is fungi. Fungi, numerous diseases in aquaculture are caused by organisms that are part of the normal biota of their surrounding environment such as fungi and fungal-like organisms or what we call Straminiphilous organisms which occur in most waters. The many known occurrences of fungal diseases in world populations and the documented devastating disease outbreaks indicate that fungal and fungal-like pathogens are important in our environment. Fungal diseases can act as major limitations on natural and cultured populations of aquatic animals. They are either saprobes that colonize decaying organic matter, or parasites to a great variety of aquatic organisms leading to epizootic diseases. So what are the major fungal diseases? First, saprolegnasis. Second, epizootic ulcerative syndrome. Third, bronchiomyces. Fourth, ichthyophoniasis. Fifth, exophiala infection in fishes. Sixth, black gillnet disease. And 7. Red disease in crustaceans. Always remember that they are generally opportunistic invaders, but once established are often fatal and difficult to treat.
Fungi constitute a group of heterotrophic organisms which contain no chlorophyll and are historically compared to plants. They are grouped by the morphology of various life stages. They are usually filamentous and multicellular, although some are non-filamentous and unicellular. The filaments known as hyphae constitute the body of a fungus. These filaments elongate by apical growth, in contrast to intercalary growth of other filamentous organisms. The hyphae are either septate or non-septate. Fungi reproduce by both asexual and sexual means, producing different kinds of spores. The fungal spore is like a seed which is resistant to heat, drying, disinfectants, and the natural defense system of fish. Mycotic infections can affect eggs, fry, fingerlings, and adult fish. Stress factors such as mechanical injury after handling, exposure to extreme pH levels, prolonged exposure to low water temperatures, lack of food, presence of other microbial infections such as bacteria and virus, increase the susceptibility of fish to fungal infections. Infection is usually restricted to superficial tissues and, unless the fish can be treated, the condition is usually lethal.
Diagnosis: potentially all freshwater, marine water fishes, crustaceans such as spinach shrimp as well as marine shellfish and abalones are susceptible to fungal disease. Detection of fungal infections relies only on the observation of gross pathology, histological examinations, standard mycological isolation, and identification procedures. Fungal diseases in general are very difficult to control or treat once they have been taken hold. Some of the treatments used are malachite green bee oxalate compound, potassium permanganate, formalin, povidone iodine solutions, copper sulfate. Prevention. Preventive arrangements are consisting of complicated set of treatments elaborated on the base of a good knowledge of the etiology of disease and a host biology. It concerns the elimination or restriction of infection sources and the possibilities of its further expansion, likewise the enhancement of condition of fish organism in the way to be able to withstand the infection. The prevention is of basic importance in disease elimination, so it is important to provide water sources free of pathogens, protection from the transfer of pathogens by uncontrolled transport of fish and spawns, disinfection of ponds, fish culture units and equipment, winter freezing and summer drying of ponds, and optimization of environmental conditions. Here are some common aquatic fungi both marine and freshwater system. Bronchiomycosis, gill rot, fungal species, Branchiomyces spp, species infected, carps, goldfish, eels. Condition for disease outbreak. It flourishes in poor quality water with high levels of organic matter. They cause a typical gill rot at temperatures above 10 degrees Celsius. The course of the disease is usually rapid and is enhanced by high water temperatures and overcrowding. Point of entry, gills. Diagnosis 1. Microscopic examination will reveal the branch and conocytic mycelia of the pathogen within the affected gill tissues. 2. Fungal hyphae in the gill obstruct the circulation of the blood. 3. The disease can appear suddenly and often has rapid course with losses as high as 30 to 50% occurring in 2 to 4 days. Death is due to anoxia. 4. Necrosis and proliferation of lamellar epithelial cells and lamellar fusions may be observed. Treatment. Various chemicals have been used to treat bronchiomycosis, which include 1. Malachite green 0.3 mg over liter for 24 hours. Benzalkonium chloride 1 to 4 ppm active ingredients for 1 hour. Copper sulfate 100 ppm for 10 to 30 minutes. Sodium chloride 3 to 5%. Note: if an outbreak occurs, feeding of the fish should be stopped and the dead fish should be removed from the ponds and buried in a lime pit. Prevention. To help prevent further outbreaks, the pond should be drained, dried out, and disinfected with quicklime.
Ichthyophanosis is caused by the fungal species Ichthyophonus hoferi. It is an obligate parasite, spherical or oval in shape, yellowish-brown and has granulated cytoplasm. The fungus lives as an intercellular parasite in the tissues of various organs. This is a fungal disease of both freshwater and marine fishes. The fungus lives as an intercellular parasite in the tissues of various organs. These parasites become encysted by host tissues. If the cyst walls become very thick, the fungus cannot reproduce and dies. If the fungus grows faster than cyst wall, it produces numerous daughter cells. Their point of entry is at skin and internal organs and different parts of the body. To diagnose the ichthyophanosis, the fish can be observed with slight or moderate infection. There are no external signs can be observed. However, the fish with advanced infections have rough or granulomatous skin. The term sandpaper effect is used to describe the granulomatous appearance. Microscopical examination can also be done on infected tissues to detect the fungi. Also, diagnosis can be done through histological examination and isolation and identification of the causative agent. In dealings with the ichthyophonus disease, there are no therapeutic procedure for its treatment. However, prevention of infection is the best method to control the disease. It includes feeding of raw fish or the infected food must be prevented. And then infected fish or dead fish must be eliminated or destroyed. All nets, brushes, and utensils used around the infected fish must also be disinfected at the same time.
So the next disease is cotton wool fungus saprolegnasis. The fungal species are cotton wool fungus, the most common presentation of water mold infection, as relatively superficial cotton-like growth on the skin or gills. So the condition for this disease outbreak. This disease attacks are temperature dependent, temperature ranging from 32 degree to 95 degree Fahrenheit, but seem to prefer 59 degree to 86 degree Fahrenheit, usually occurring at low temperature. That is for the condition for that disease outbreak. The diagnosis for this cotton wool fungus, or what we call saprolegnathis. First, a fish suffering from saprolegnathis will exhibit cotton-like growths on the skin and gills, depigmented skin, and sunken eyes. Second, in more severe cases, the cotton-like growths can extend into the muscle tissue. Infected fish will begin to die slowly over time. And the third one, the area of the skin and gill damage determines the severity of the disease. The clinical symptoms for this cotton wool fungus, or what we call saprolegnathis, the fish fungus appears as gray or white patches on the skin or gills. They may become brown or green, which is the later stage as they trap sediment. Saprolegna normally establishes as a small, focal infections that then spread rapidly over the body or gills. So, what should be the treatment for this disease? Fish are removed from the water. They appear to have a slimy matted mass growing out of the skin and scales. Use the 100 milligram per liter strong malachite green solution to clean the lesion and apply a waterproof cream. So the prevention for that is it can be prevented by avoiding rough handling, crowded stocking conditions, and poor water quality. It should be prevent further spread and reduce overall mortality. Fish should be harvested as soon as saprolegnathis is observed.
Black gill disease, Fusarium disease. Fungal species. Black gill disease is caused by an Ascomycetus fungus Fusarium solani and probably other species of the genus including F. moniliform. Species infected. The disease can affect all pinnate shrimps, example, Penaeus monodon, Penaeus vanamae, and Penaeus japonicus during pond grow-out stage. Point of entry, gills. Condition for disease outbreak. In aquaculture settings, ascorbic acid deficiency is also known to evoke black gill in pinnate shrimps, usually associated with poor pond bottom conditions. Diagnosis. Number 1. Microscopic examination of wet mounds of infected tissues will reveal the presence of canoe-shaped macroconidia. Fusarium spp are ubiquitous soil fungi. 2. Infection may begin at different loci and spread slowly. 3. Fusarium solani is an opportunistic pathogen of penates and are capable of establishing infection in shrimp compromised by the other stresses or overcrowding. 4. Appearance of black spots in or general discoloration of the gills due to melanization at sites of tissues necrosis that precede mortalities in juvenile shrimps grow in ponds. Treatment. No treatment is available for fungal infestation without harming the shrimp. Prevention. Number 1. Fusarium infections are difficult to pharmaceutically treat in infected individuals. 2. Preventive measures include the good management of the pond bottom, elimination of sources of Fusarium conidopores, and destruction of infected individuals. 3. Several fungicides show promise in vitro but once proved to be effective in actual field trials.
Hi, thank you for watching our video. We are the Group 1 from BSF 3-1.
I am Vladimir Dandan and here's my co-narrator.
Hi, I am Ryan Deliota.
Hi, I am Danielle Fakud.
And of course, we will not be able to complete this presentation without the help of our team. May we introduce them to you?
Our researchers who gathered the necessary data used in this presentation.
I am Lailani Siblawan.
I am Virgaliala Ansi Fakun.
Hi, I am Cherry Jane Santiago.
And I am Jennifer Mercado.
And of course, with the help of our audiovisual team, I am Mary Jane Manabat.
Hello, I am Emery Felicity Garland and I was in charge of summarizing all of the needed info from the researchers and creating a script.
Hi, I am Ryan Deliota and I gathered the pictures and background audio for this video.
Hi, I am Danielle Facud and I made this PowerPoint presentation by using the script as guide.
And of course, our editors.
Hello, I'm Patricia Demir I. Magnase. I was the person to gather all the files needed and did the preliminary editing for this video.
Hello, my name is Erickson Magdalam and I did the polishing edit as well as added the subtitles for this video.
Thank you for listening and we hope you learned something.